Basic biology of plasmid and phage vectors. So, to deliver the target DNA into the host microorganism, we need cloning vector or vehicles so that the target DNA, after insertion into the host organism, it can replicate there and express there. Scientists has developed different type of vector system with the passage of time, and they may be of different types. Like the bacterial plasmids, they are well famous, and bacterial plasmids, they can deliver the target DNA that is having the size of 15,000 base pairs or 15 kilobases. After plasmids, even lambda phages have also been used to deliver target DNA, and they relatively transfer larger DNA fragments as compared to plasmid. And the size of the target DNA, it may be 24,000 bases or 24 kilobases. Then some plasmids that contain the cohesive sites of the phage vectors have also been developed. And those plasmids that contain cohesive sites that are necessary for the phage particle packaging, they are called as the cosmids. Now these cosmids, they can transfer target DNA having the size of 34 kilobases to 47 kilobases. And some other vectors, that is phagemids or phasemids, they have also been developed from F1 phages. तो बाज़ दफ़ा क्या होता है कि फ्रॉम यूकैरियोटिक सोर्स आपको ज़्यादा बड़ा फ्रैगमेंट जो है वो क्लोन करना पड़ता है. That is not possible by using plasmids or cosmids or lambda phage vectors. So, under such situations, scientists have developed other artificial vectors like bacterial artificial chromosomes or bags that can deliver larger DNA fragments. And even to clone larger DNA fragments in yeast artificial chromosome have also been developed. But we are going to start our discussion with plasmids. As you are well aware that plasmids, mostly they are circular DNA molecules that can replicate independently of the host chromosomes because they contain their own origin of replication, so they are called as replicons. जैसे मैंने कहा कि प्लाज्मिड्स जो हैं ये मोस्टली सर्कुलर होते हैं, लेकिन ये हमेशा ऐसा नहीं होता. There are some examples of bacteria like gram-positive streptomyces or even some spirochetes like the Borrelia. They also contain linear plasmids, but because they have their own origin of replication, so that's why they are also called as replicons. But overall, plasmids, they are circular DNA molecules. With reference to the size of the plasmids, their size, it may ranges from a few thousand base pairs to 400,000 base pairs. They may be having low molecular weight or high molecular weight. This diagrams indicates the presence of nucleic acids in prokaryotic microorganisms like a bacterium. So the portion of the chromosomes that occupy the cytoplasm, it is called as the nucleoid. So it is very famous region in bacteria. And in addition to the bacterial chromosomes, some other type of nucleic acids are also present, and they are well known, and they are called as the plasmids. Now, the number of plasmids, in the cell, it may be variable, and it is commonly called as the plasmid copy number. And this number, it is variable from one species to another species. Now, the foreign DNA can be inserted into the plasmid vector. So, artificially recombinant plasmids, they are produced. How it can be inserted? You are well aware that this uptake, it may be mediated by certain chemicals, that is in the presence of calcium chloride solution. And sometimes the cells, they are natural competent, that is they naturally have the ability to take the foreign DNA. But sometimes this competency, it is artificially induced, and for this purpose calcium chloride solution, it is used. So artificially created recombinant molecule like the plasmids that contain the foreign DNA and the host microorganisms, they are incubated at low temperature, and then the temperature, it is suddenly raised to 37 or 43 degrees Celsius. So it will temporarily increase the permeability of the membrane, so that the artificial recombinant molecule, it is being taken by the host organism. Another technique for the insertion of recombinant molecule, it is the use of electric pulse, and the technique, it is known as electroporation. Now, if the bacterium, they contain plasmids, what advantage it can enjoy by having such extra chromosomal DNA or the replicons? So briefly at this point, I can say that when plasmids are there, they increase the bacterial capability to survive in the environment, especially when some sort of stress, it is present. So it increases the survivability of microorganisms under natural conditions. But in addition to that, it can also exhibit different beneficial traits that we are going to discuss after some time.